The overall goal of this procedure is to isolate and digest bacterial cell walls to determine the identities and relative fractions of different muropeptides using UPLC analysis. This is accomplished by first lysing bacterial samples by boiling in sodium dodecyl sulfate. After washing out SDS, the second step is to digest samples with pronase E to purify away the outer membrane of gram-negative bacteria. Next, the samples are digested with muramidase to solubilize the peptidoglycan into individual muropeptides. The final step is to reduce the muropeptides and adjust pH to the muropeptide isoelectric point. Ultimately, UPLC is used to separate muropeptides according to size and hydrophobicity, facilitating the quantification of important cell wall characteristics, such as degree of cross-linking and average glycan strand length.
This method can help to reveal the answers to key questions in the field of microbiology, such as the relationships among morphogenesis, cell wall architecture, immune system activation, and pathogenesis.
Although this method has been developed to purify gram-negative peptidoglycan, it can also be applied to gram-positive bacteria with the addition of a few enzymes and chemical treatments.
To grow the bacterial cultures, back-dilute overnight cultures 1 to 100 into 250 milliliters of fresh media and grow to the desired optical density at 600 nanometers.